You know,
Okay,
almost.
She has changed the hair. She's changed the hat now suddenly.
Yeah, it's a sudden change for me.
Yeah, yeah.
So what are we calling this talk?
You have that with you.
I'm just doing this.
here you have.
So I will let you know. So he has invited long back. That's why today also I invited.
Dr. Amitabh, what is the topic title?
So dermatology and genomics.
So we'll just say dermatology and genomics, or
Understanding pathology at molecular level.
dermatology and genomics.
Shall I send it to you now? The flyer.
No, I'll just scribble it.
Dermatology and genomics.
Understanding pathology at the
molecular level.
And have you been working in dermatology, Dr. Amitabh, or
yeah?
You have to unmute yourself.
Voice is not coming.
Yes. Now.
Now.
Actually, I am working as a genomist, not
Yes.
not in terms of dermatology. So I am expert in a genomics part.
Yeah. So, I mean, we just to explain the title, why are we talking about dermatology? Because they will be interested.
So my topic is actually next generation sequencing, its application in clinical dermatology and diagnostic.
Application of NGS sequencing.
Yes.
Application of NGS in clinical dermatology and diagnostic.
Correct. Diagnostics. So that will be a follow-up of Dr. V.R. Rao, who will be talking the deep technicalities about the dermatology and genomics.
Rare skin diseases.
With case studies and all that. I think Professor Rao, I think you have to you give ample case studies of what we have done preferably, or what are the best that can be shown.
I have just sent you the flyer, madam. WhatsApp.
Yes. So I would like to give a brief introduction to the Genome Foundation.
Yes.
The Genome Foundation is one of the very few non-profit genetic testing and
genetic diagnostics, research and training.
Genetic testing, diagnostics and
diagnostics,
research and training centers in with ideals of the founding fathers.
I will also give anything. I will also talk a little. You can just give one line.
I'll just give a very short introduction.
Yeah.
Is one of the very few non-profit genetic testing, screening, testing, diagnostics, research and training institutes in the country, striving to maintain the ideals of the founding fathers. So this foundation was set up by Doctors PM.
Dr. Gandhi will be talking about.
Chapman, sir?
Dr. Gandhi will be introducing about Genome Foundation.
I'll just give one line.
Yeah, give one line. One line.
One line, because why this
Correct.
Why
Yeah,
this
correct.
session and
Yeah.
why this topic? Just that
Okay.
much. Very brief. All my introductions even will be very brief.
Okay.
Yeah.
Silata, have you joined? Silata.
No. Okay.
Somebody's here in Genome Foundation name.
That's Karan or
Kanna. Are you there, Kanna?
Power,
Kanna, you are there.
sir, Srilata power is, sir, power ledata.
It's at home. He's at home now.
Yes, yes, I am there.
warm welcome to this evening's session, which is on dermatology, which is on the subject of dermatology and genomics.
So, Pari, I think you got the full introduction of full profile of Dr. V.R. Rao. Dr. Pari.
Yes, I have it, sir. I've just printed it out. So, how much shall I speak? I thought I'll give a brief introduction.
Yeah, about 2 to 3 minutes, or maybe 4 minutes. Since he is the main speaker, people should know what he is doing.
Oh, I thought Dr. Amitabh Biswas was the speaker. I didn't know that Dr. V.R. Rao is the main
No,
speaker.
Professor VR is our hero today.
Yes, that I had to.
Amitabh is not a villain, but he's a side hero.
Yes. Oh my. See that this was why I was connected.
So, and all that today talk is going to be centered around Professor V.R. Rao, who has been doing a lot of biodiversity projects for the last 50 years.
Yes.
Heading the Anthropological Survey of India as its director, and he has traveled extensively throughout the length and breadth of the country to understand the biodiversity, and in the process he found a lot of new and rare diseases, and ever since he found that he took special interest in that, and then Dr. A.S. Kumar, good evening. Yeah, he took special interest and then trying to do a lot
Good
of
evening,
work.
sir.
Good evening, how are you? Yeah,
Fine.
Yeah. We are just trying to log in, waiting for others to join.
Yes,
So
sir.
Dr. A.S. Kumar, he's not seen, but I can introduce Professor V.R. Rao. Professor V.R. Rao is seen in blue shirt. And of course, you can recognize me, and you can recognize Amitabh Biswas. He's a new age bearded hero. Because we can't see beards in our age, so he's a new age bearded hero. And then you see a lady, Pariplavi Mokkapati. She was professor of anatomy in Usmania and Gandhi Medical College. She's going to introduce the speakers today. And slowly others are joining.
Okay, sir.
Dr. A.S. Kumar also is a avid researcher, and he has done a lot of research work. I know his work when I was the ethics committee member of OIC Hospital. So I must have seen more than 10 different research papers of him. Only thing is, I can't make out technically what they were, but that's why I continues to be with. Yes, Mr. Priya. Good evening, Mr. Good evening, Professor Mr. Priya. How are you? Nice to see you. Nice
Hello,
to see
ma'am.
you. Professor, I think Pariplavi, you are able to recognize Pariplavi, Mr. Priya?
Vishnu Priya, ma'am.
I think she's frozen.
She's here.
Professor Mr. Priya, I think you are able to see Pariplavi.
Yes, I'm able to see her.
Yeah.
I know.
She is a new addition into Genome Foundation.
Yeah, I know. She's our department student. Like she did her PhD in our
Graduate
department.
from there.
I see. Okay. Good. Good to know, yeah. So how is everything? You are doing fine. What is the time now?
And now morning 7:30.
7:30 is okay. I think we didn't wake you up too early.
No.
So when are you likely to come in this year, or you want to wait? Huh?
Actually, I'm planning to come in July, but again the cases are going up.
Oh yeah.
So, but still, I think the proposal is for July right now.
I see.
Mm.
Okay.
But we didn't buy the ticket yet.
It's all right, but we await your arrival, and we'll give you good welcome when you come back.
Thank you so much. I would like to see all of you.
Yeah. Okay. Slowly people are joining now, and whoever is going to start off, we'll give some time. Meanwhile, we can be talking to each other. We'll give some time because Indian Standard
I
Time
think
is not.
whoever is admitting, Mr. Karan or Srilata, whoever is admitting, we can, okay, it's already time, we have to admit.
Karna, are you admitting everybody? Karna, are you admitting everyone?
Yes, sir.
Are you admitting everyone now?
Yes, sir.
Yeah.
Everyone is coming, sir.
Okay, now you can continue. Now the screen is about 12 now. So I think you have the full screen. We'll start working because let them join a little later. But one announcement you have to give that you have to keep all the telephones mute. So it doesn't look nice if I give the announcement. Pari, I think you can give announcement when you start. So let all the telephones be in mute, and and also when they are not talking, when the speakers are talking, let them mute themselves. So these are the two instructions. I think.
Yeah, I'll do that.
Yeah.
By the way, I would like to congratulate Professor V.R. Rao and you, sir, for starting a nice program. It's a very good initiative.
Yeah. Yeah,
It's
good.
good
I think I
I'm
would
very
have
happy.
I would have been still in inertia. Professor Raghavendra Rao has woken me up.
Yeah.
And now I want to make it, if not a fortnightly future, monthly future at least.
Yes, it's
And
something like.
Yeah, and we are waiting for you to come and then
Yes, sir.
fill 1 month of socket.
Yes, sure, definitely.
And Amitabh Biswas with his knowledge in NGS, he is going to add a lot of value to our lectures, especially a module in NGS with relevant topic covered will add value to all our future programs. So that's what we are thinking. So sure that Amitabh will be able to make himself available for our requests.
Yes, sir, definitely, sir.
Yeah. Srilata, are you there? Sekhar is there. Sekhar, we're going to record this, Sekhar?
Good evening, sir.
Good evening, Srilata, yes. So Karna, are you going to record this?
Recording, sir.
Recording, okay. How about the Facebook and YouTube account? Somebody
Yeah,
has that.
YouTube is activated. In 2 minutes, the Facebook also is activated.
Okay, good, good, good. So to get proper bandwidth, we can say, let these, except the speakers, let others be off from the screen. But means the video can be switched off. After starting the program, after introductory remarks, and after the first speaker, after I speak, and after VR starts his speech, after his introduction, you can say let only the speaker be available on the screen, and others can mute themselves on the video, both video and audio. So this can be one of the, I think, house instructions by Pariplavi.
Yes. Yes, I will.
Yeah.
Or wait for a few minutes more?
6:02 now. Let us
Yeah.
give, for Indian Standard Time, I think 5 minutes is okay. Beyond
Yes.
that, it doesn't look nice.
Yes.
5 minutes, just we can fast start off within 5 minutes. That means within next 2 minutes, we can start off.
Yes, sir.
Dr. Diksha, good evening to you.
Good evening, Gandhi Garu.
How are you?
Yeah, fine.
Happy that you are here, and together we are participating. I am not a geneticist, I am not a medical doctor, but still, from
But you
Genome
are
Foundation.
doing a great service to dermatology. That's what I think. This is a very important CME.
Thank you very much, and I'm sure that we'll be able to work together from now onwards through this forum. And Dr.
Sure.
I.S. Reddy, also I know personally, my grandson when he was born, I used to take him almost every day to Dr. I.S. Reddy. Welcome,
Oh.
Dr. I.S. Reddy.
Very nice.
Yeah. So happy to see some of the names. Okay, now I think time now. Dr. Pari, I think Dr. I.S. Reddy, yeah. Dr. I.S. Reddy, thank you. So you may not recognize me.
Yeah, I know, sir. I remember you very well. Yeah, I remember you very well. You were in the you were used to head the forensic department, the forensic wing of the.
Yes, state government. Yes.
Yeah, I remember you very well, sir. Yeah.
Thank you. Now I am wearing the hat of Genome Foundation because of circumstances. I will explain that quickly when I start my session. Thank you for being with us, and we look forward to have your continuous association from now onwards. Now, Dr. Pari, I think we can start off now.
Yes.
6:06.
Good evening, ladies and gentlemen. All the elders, all the distinguished scientists, medical professionals, students, researchers, clinicians, and other guests who have all come together this evening to participate in this wonderful and exciting session in different capacities. A very warm welcome to one and all. I am Dr. Pariplavi, one of the directors of the Genome Foundation, welcoming you on behalf of all the members of the Genome Foundation. Now, what is the Genome Foundation? The Genome Foundation is one of the very few nonprofit genetic screening, testing, diagnostic, research, and training institutes and centers in India. and it strives to keep up the ideals of service, of science in the service of the society. It was set up by Dr. P.M. Bhargava, Dr. Lalji Singh, Dr. K.P.C. Gandhi, and some other distinguished scientists. I will have Dr. Gandhi talk about it in a short while. So the speakers for this evening's session are Dr. K.P.C. Gandhi, Dr. Badlamudi Raghavendra Rao, and Dr. Amitabh Viswas.
Your room is.
Genome Foundation also has, amongst others, in the service of society, distinguished scientists like Dr. Radha Ramadevi, who was heading the pediatric and neonatal screening of the newborn in the Center for DNA fingerprinting and diagnostics. We have Dr. Kanakabhushanam, the former director of health services of the combined state of Andhra Pradesh, and other distinguished scientists like Dr. Nalini Gupta, Dr. Zredanna of the Central University, who are all
Professor Vishnupriya.
Doctors Vishnupriya, yes. Welcome, madams and sirs. Also present is Dr. Srilata, who is the quality control manager and chief of the lab at the Genome Foundation and the Truth Labs. So let me now proceed to introduce the speakers of the evening. Managing Director of the Genome Foundation is Dr. Gandhi P.C. Kaza. Not many people need an introduction to Dr. Gandhi. He is the Sherlock Holmes of India. And I have been advising some of our medical students just recently to read up about Truth Labs and about Dr. Gandhi, who is the advisor to the governments of Andhra Pradesh, governments of West Bengal, the Jammu and Kashmir, in the Department of Forensic Science Services. He's the chairman of Truth Labs. And I'll give a very brief introduction. He has trained in different centers in India and also in New York City. Dr. Jawahar, could you please mute your phone, please? Dr. Jawahar Gandhi.
Jawahar Gandhi, I think you have to mute.
Yeah.
You are heard. Jawahar Gandhi is heard.
Facebook live it is.
No, Jawahar Gandhi.
YouTube kuda.
Dr. Jawahar Gandhi. I request all the participants, all the attendees to make sure that their audios are muted, and preferably as the session proceeds, to save bandwidth, even the videos could be muted.
Ma'am, host can mute anybody. Host can
yes,
mute
host can do that.
Host should do these things.
Yes. Karan, will you please see to it? Thank you. So during his long tenure as the director of the AP Forensic Science Labs, Dr. Gandhi brought in several radical reforms, which made it one of the model training institutes, not just across the country, but even internationally. Because he himself has undergone training and development in very premier institutes of the world, such as Interpol, Lyons in France, Scotland Yard, London, then the Bruns Criminal Land in Frankfurt, Germany, in Ottawa, in Tokyo, and a number of other places. He also facilitated the AP Forensic Science Lab, being the first government department in India to get the ISO 9001 certification in 1999, and later the first forensic lab to get the ISO 17025 NABL accreditation also. And he has played a very pioneering role in forensic science education, having helped set up the Masters courses in criminal justice in Andhra University, the master's degree in forensic science in Osmania University, the PG diploma course in criminal justice and forensic science in the University of Hyderabad, and the master's degree course in forensic science in the Banaras Hindu University also in the year 2014. And now the PG diploma course in criminology, criminal law and forensic science at Nalsar. So that is really very, very impressive. Even I did not know that until recently. And it's a very remarkable achievement in the field of forensic science education. Dr. Gandhi has been responsible for the training of more than 10,000 senior judicial, police, prosecution, banks, insurance agents, corporate, legal, vigilance, and security officers across the country. Exclusively organized, these training camps have been exclusively organized by the Truth Labs in the last 10 years. Needless to say, Sir has won a number of awards and accolades and distinguished memberships. Truth Labs is currently handling cases referred by various courts, high courts, the Supreme Court, a number of police departments, the CBI, the NIA, banks, public, private, foreign banks, insurance companies, MNCs, corporates, law firms, detective agencies, and the ordinary citizen who can just walk into the lab and seek help in obtaining justice. So the courts of 20 states, the police of 18 states, all the 50 banks, all the 30 insurance companies, over 100 central and state government organizations, over 1,000 MNCs and corporates, individuals of various Indian states and foreign nationals of over 20 countries have sought the services of Truth Labs until today. We are very proud and privileged to have you on this occasion, sir. Thank you so much for coming.
Thank you.
We have another luminary from the field of genetics, molecular biology, and service to society. None other than Dr. Vadamudi Raghavendra Rao, whose education has been at various places from the VR College, SV University, Nellore, from SV University, Tirupati, Andhra Pradesh. He did his MSc and his PhD from the KEM Hospital in Mumbai. Dr. V.R. Rao has traveled extensively across India as an official of the Anthropological Society of India, which he was heading as Director General in charge for a long time. And during this course, he did a great deal of pioneering work studying the diverse ethnical groups, the tribes, the lifestyles, and most importantly, preparing registries. We'll get an idea of his work when we look at just a peep at some of the aspects of his, curriculum vitae. ICMR in different institutes, the Institute of Immunohematology. He worked as grade 4 scientist at CCMB Hyderabad, as joint director, director of the Pathological Society of India, as professor, biochemical genetics, molecular anthropology, University of Delhi. He is the ICMR Emeritus Medical Scientist at the Department of Genetics of Osmania University, which, as we all know, has a very strong Department of Genetics. He is currently the research director at the Genome Foundation. Sir is a life member of a number of professional and scientific bodies and societies, like that of the Medical Anthropology, the Bombay Hematology Group,
Under the introduction, Society,
He's a member on the Committee on Health and Drinking Water constituted by the DST. Some of the books that he has been involved with writing, these give a glimpse to his enormous expertise in these areas. The Genetic Atlas of the Indian Tribes. Hemoglobinopathies in Indian populations. This is a series in bibliography on the human genetics in India, which he has co-authored with other scientists like Dr. S.R. Naik and Dr. Mukherjee. The third book, Human Origins, Genome and People of India, Genomic, Paleontological and Archaeological Perspectives. I'll repeat that, it is so impressive. Human Origins, Genomes and People of India, Genomic, Paleontological and Archaeological Perspectives. Another book is Identity, Cultural Pluralism and State, South Asia in Perspective. Identity, Cultural Pluralism and State, South Asia in Perspective. Sir has done enormous work in the field of hemoglobinopathies, thalassemias, sickle cell anemias, and in the screening and diagnosis, counseling, and other aspects of the health and well-being of various ethnic groups across the country. We are very, very privileged to be able to listen to Dr. V.R. Rao this evening. A very warm welcome to you, sir. Distinguished scientist of this evening is Dr. Amitabh Biswas, who is presently working as a consultant in genomics with special reference to whole genome sequencing and also coordinating and managing the project and data analysis. He is involved in planning and organizing the work, training, the training part of it, and guiding the team to optimal sample management and guiding the scientific experiments. He had also worked on the genetics of cardiomyopathies using next generation sequencing tools for whole exome targeted sequencing in familial studies. He has over 10 years of experience in clinical research setting, and he has various research projects. We have people with so much of education, research, and other work behind them that the few minutes that we have to introduce them are not enough. So I do not wish to stand between the speakers and the audience. So once again, a very warm welcome to all the participants in today's session. A small request, repeat, please make sure that your phones are on the silent mode, your audio is muted, preferably your videos also, unless later on you wish to speak or interact with any of the speakers. Thank you all very much and welcome, sirs.
Good evening, everybody. I being the first speaker to just say a few remarks about what this series of programs which are going to have from now onwards under the auspices of Genome Foundation. I just wanted to say a few words about Genome Foundation. Genome Foundation is a brainchild which was born when we were just sitting in our ante room, both myself and Dr. Lalji Singh. We have something in common. which many people may not be aware. Lalji was called back in 1987 end by Government of India when he was in Commonwealth Fellowship, saying that his presence is needed to do some advanced molecular genetics work, including DNA fingerprinting. And I have been called back by the state government when N.T. Ramarao was the CM, when he started the first state-level police academy in India, in Andhra Pradesh, saying that we need your services, you have to come back. When at a time when I was recruited by New York City Police for appointment as a civic investigator of ethnic groups, especially when there are a lot of violence going against South Asians in New York City. Both of us landed almost at the same time in 1987 December, and both of us, having known about the development of the DNA fingerprinting by Alex Zafri in the UK, we thought we should do something together. And he started developing the DNA probe for DNA fingerprinting in CCMB. And I gave samples from the police department to the police academy and later on to the forensics laboratory. That has brought us together, and the friendship continued till he left us in 2017 December, unfortunately due to heart attack. And he was admitted in the hospital next to the Banaras Hindu University, Banaras Airport, Varanasi Airport, where he has set up as Vice Chancellor of BHU a trauma center. And he died in the trauma center, which was set up as a part of BHU's outfit. It's very unfortunate. We missed him. Then, and how we thought of this Genome Foundation was when we were sitting together, when Human Genome Project became possible. So both of us were discussing, although Human Genome Project can bring a lot of developments across the world to the human population, but for Indian population, how soon they'll be able to see the advantages, fruits of this genomics. So then both of us felt it may be difficult for the government alone to do it. It may be difficult for the institutions to do it. Let us do as a social service from our side, as a part of our post-retirement non-profit work towards continuous responsibility to the nation. Both of us being patriots and nationalists, we just started Genome Foundation as a non-profit company supported by CSIR when Dr. Machelkar was the CSR Director General. It was a starting point, and we have gone through very many ups and downs. Ups and downs, I mean. And when I retired to take up something like this, I had to start Crime Stoppers Foundation, and later on through Truth Labs, which have become too busy with 6 Metropolitan centers across the country. And he became busy after he took charge as BHU Vice Chancellor, and ultimately it has to take a second back seat. And only in 2016-17, when he came back from BHU, he started working on Genome Foundation seriously. So he did work a lot of things, and during that period, and we have done a lot of work of screening the people in his own native district in UP, close to Varanasi. And then a lot of work has been done in Telangana population during his time in CCMB, and later on as a Bhatnagar fellow also. But because of his sudden demise, the responsibility fell on me, because the chairman, Dr. Rangarajan, former governor in RBI and former governor of Andhra Pradesh, he said that a non-profit organization, nobody would like to manage it, and only you, the co-founder, has to do it. And I took the responsibility, and in the past 2-3 years, we tried to stabilize the operations, and today we are able to screen the patients who come to us. About 40 different diseases have been so far identified in the patients coming from various parts of the country, and especially from Telangana region and neighboring states also. And Genome Foundation is being named as a people's organization. It is meant for people's welfare, but it operates on non-profit basis, charging only uh, the the cost required for differing costs for operations. That's why in many of the cases, the charges that which we charge for the genetic screening is 1/3 or sometimes 1/4 of the total cost charged by other organizations. So this being the specialty of Genome Foundation, and since after I took charge, I invited very senior experts on voluntary basis. Please listen to this carefully. On voluntary basis, those who retired and those who got aptitude to do some good work, continued good work like me and Lalji Singh and a few others. And Professor V.R. Rao is one of the first few people to join us on voluntary basis in Genome Foundation to continue his research interests in rare diseases. And similarly, Professor Priya, whom we have seen, and Pariplavi, and Radharama Devi, and Dr. Suma Prasad, and also a few others like Dr. Nalini Gupta and Dr. Kanakabhushanam. And these are all the few people whom, and Dr. Professor Reddanna from University of Hyderabad. They have been voluntarily doing their services in Genome Foundation and providing services to the needy patients. And so far, in UP, Andhra Pradesh, and Telangana, in neighboring areas, we identified the common genetic diseases like beta thalassemia, sickle cell anemia, Down syndrome, and rare diseases like Duchenne muscular dystrophy, and some skin diseases like Malde Melade, acropigmentation Kitamura, Job syndrome, and a few others like familial Mediterranean fever, hyper IgE, total anomalous preliminary venous return, hemophilia, primary immunodeficiency. Sorry if I have not pronounced the names well because I am not a geneticist. I am a PhD in physics and criminal justice, but I don't have much knowledge of genetics. But I am trying to learn something about genetics. Of course, I know basics of genetics and remaining things. And Professor V.R. Rao will be talking to you. And so far, we have produced more than 10 research papers. And on an average, we receive 10 patients a day as of now. And we are trying to extend our areas of operations, of coverage to rare diseases, not only on the dermatology sector, some cardiomyopathies and newborn screening and prenatal screening, and things like mentally retarded cardiac, pregnant woman, newborn or non-invasive prenatal testing, pre-implantation genetic screening in IVF cases, karyotyping for causal analysis of recurrent abortions and all these things. These are the few things which we are currently engaged. We want to extend it to several areas, and I invite all of you to please join us in our giant efforts to serve the people voluntarily as a part of our social responsibility and national responsibility. And I on my own behalf, I invite every participant who has joined us in this evening, on Sunday evening, for going their little weekend time for the cause of nation, for the cause of the community around us. Thank you, all of you, and
Thank you, sir.
yes.
We are really inspired. It is really impressive and very inspiring to be associated in causes such as these with people like you involved. I would now like to invite Dr. V.R. Rao to address us. He has the expertise, leadership, training, and motivation necessary to conceive and successfully implement scientific projects. Besides the teaching and administrative assignments that he has done for over 35 years, and with a broad background in human genetics and specific training in genomics of complex diseases, human genome diversity of Indian populations, and his research as principal investigator funded by ICMR, DBT, DST, including application of whole exome sequencing in cardiomyopathies, complex genetic basis of suicides, and the association with other psychiatric traits, the endophenotypes, and lifetime perceived stress factors, with work in the genetics of Parkinson's disease. With all these, he brings with him a very large repertoire of skills and knowledge and expertise. With his diversity studies on the Indian populations led by Dr. V.R. Rao, provided massive empirical genome-based evidence of 27 primitive tribes. And this has led him to infer that our populations harbor ancient DNA signatures dating back to 50,000 or more than 60,000 years before now. These findings are found to be the basis for evolutionary understanding of disease genes and processes. He has also successfully administered projects, staffing, research, protections, budget, etc., and collaborated with numerous researchers at national and international scale. So we are very honored to have you this evening, sir, and honored to be able to listen to you. Very warm welcome to you.
Thank you, madam. Can I share my slides, share my screen? No? Are you able to see the screen, madam?
Yes, we can.
Yes, okay.
The sound is also good.
Yes.
Yes, madam, yes, madam. This is the title of my talk. In fact, the major theme of my presentation, is we have an opportunity in rare skin diseases. Okay, rarely we get this opportunity in several other diseases, wherever we do genetics. Here is a situation where 80% of rare genetic diseases, skin diseases, are monogenic. Okay, single gene causing the disease. And also, we have a situation 100% penetrant causative mutation. The mutation which we find in any one of these genes, one gene for a disease, the mutation is highly penetrant, 100%. Rarely we get this situation when dealing with, even with beta thalassemia, where we know it is a monogenic disorder, but there are several other factors influencing, okay, epigenetic factors like that. But here I find, this situation here in rare skin diseases, the opportunity for patient care, okay, family counseling and prevention. Because we are sure about the mutation and we are able to counsel patients, in terms of prevention. At the same time, translational medicine, big opportunity, enormous opportunity. All over the world, things are already taken off. They have gone a big way. Looking at the opportunity, they have gone a big way in translational medicine. This is my major theme. But to understand the theme, we need to understand the background, What is human genome at a glance? How many genes humans have? It is a very complex at looking at, it's a very complex phenomena. But at the same time, I can derive that there is some simple things like, what I am talking about, homogeneity disorders. Genetics testing approaches, okay? Lot of the technology has changed, And even the simple techniques, you can help the patient. That is what my issue here. And genodermatosis. There are several genodermatoses and broader classification has been given. But under that, what we are interested is rare genetic diseases, okay? And define what is rare genetic disease? Definition, And people started listing genes, listing these rare genetic diseases. There are several databases. I'll be talking all those things, and also translational medicine. But when I'm explaining, this is the complexity, how it has come to simplicity. Ultimately, I will take an example of a monogenetic disorder, that is rare skin disease. Let us first look at the human genome at a glance. I'm going to show you one video. It's only 4 minutes. Have patience, and you'll understand.
Life, a sunny day, a simple friend, and a complex biological story. The Human Genome Project is a way of exploring our molecular selves. Almost all of our cells, the muscle cells that let us smile, the brain cells that perceive the humor in things, the cells of our eyes that take it all in, contain a complete set of all our genes, the genome. If we could journey inside ourselves into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule, messenger RNA. The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus, but messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA to make a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. tRNA molecules help translate the language of DNA and RNA into the language of proteins. tRNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the laborers. Some form structures like tendons and hair. Others perceive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular selves, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personalities, and risks for diseases like cancer and heart disease. A growing understanding of our genes and all they do will help us understand the complexity and the wonder of life. A simple friendship, a sunny day, life.
Highly orchestrated biological process. It's a universal biological, all life beings have this process. Look at the enormity. Humans, we have 37 trillion cells. And each cell has got 23 pairs of chromosomes. And in each chromosome, we have what is called gene, okay? The nucleotide, DNA molecule is a linear molecule on the chromosome, okay? And this molecule, each chromosome has got 3 billion base pairs. Base pairs means nucleotide sequence. Each base is a nucleotide, and 3 nucleotides form a protein. Okay, the nucleotides on the chromosome, they're linearly arranged. There are 3 billion. One pair of chromosome has got 6 billion base pairs. Okay, enormity of information and highly specific process and complex, but at the same time simplicity because it is very specific process. Okay? Anywhere, any change, that's what we call mutations. It is expected after the Human Genome Project was completed, after they sequenced 3 billion base pairs, it was expected that there will be around 1 lakh or more genes in human genome. But ultimately, they found only 22 to 25,000 genes. The length of each gene ranges from 800 in alpha thalassemia, it is 800 to 20,000 genes, 20 lakh genes. 20 lakh genes, 20 lakh base pairs, each gene length. 20 lakh base pairs in Duchenne muscular dystrophy, DMD. That is the range, of each gene. Now the question of how many genes in human genome, there are several databases. What we are until now interested is in protein coding genes. Okay, this is of our interest. What we call mutations is because of nucleotide changes, a protein is being changed, so we have a disease. And they range from 19 to 21,000 like that. But the complexity lies. Complexity means it's not so simple. There are several other non-coding genes, long non-coding RNA genes, antisense RNA genes, miscellaneous RNA, pseudogenes. There are several, Until recently, these things were thought to be junk DNA. But now it is being revealed that they have function. Okay, that's why genetics is being complicated in certain diseases, but there are still some simple diseases where the things are very simple for us. But as per the complexity, the technology is also developing now. We have several other NGS platforms, even we need to, you can, in fact, duplications, all these type of variants can be found. So many variations, mutations, types. Single nucleotide, one nucleotide change is the mutation, what we call now. Usually we type this, and also deletion, insertions, 2 to 3 nucleotides or 4 nucleotides, 5 nucleotides. There is a deletion, there is an insertion, there is tandem duplication of repetitive sequences, copy number variations, translocations. There are several types of variations. Okay. But unfortunately, for several monogenic disorders, we are still in this simple nucleotide variation situation. Okay? Genetic testing, there is a paradigm shift in clinical practice. Until recently, treatment is based on symptomatic. But now more interest in causative. We need to know cause of the disease so that the side effects of any drug, any symptomatic drug which we take is reduced. In knowing the cause, the DNA is going to help a lot, DNA analysis. And basic research also, there is a paradigm shift. Mendelian phenotype, what we are talking Mendelian phenotype is the simple disorders, rare skin disorders or Mendelian phenotypes, to complex phenotype. Some Mendelian traits like beta thalassemia, ultimately what we are seeing is a complex phenotype because several epigenetic factors. And until recently, complex phenotype means several minor mutations contributing to the complex in interaction with the environment. That is what the complex phenotype and common variant. But now, because of technology, what we are trying to understand is, though it is a complex phenotype like type 2 diabetics, there may be several genes interacting with the environment and causing the disease. Ultimately, now we are understanding there may be some rare variant, okay, rare driver mutation with 100% penetrance, with a lot of penetrance. It is a driver mutation, okay. Now, all emphasis is now going towards rare variant analysis, whereas in Mendelian traits, we are direct with rare or mutation analysis. So there are opportunities here for some, especially for rare skin diseases, but in complex diseases also, there is going to be this type of situation where single driver mutation may be responsible. In approaches also, technological approaches, there is paradigm shift. Conventional genetics, what we used to practice genetics. If I want to know the disease, phenotype, I want to know the disease, what is the genetic basis for a genotype, then I go to families, large families or populations, and do linkage study with one marker. Suppose ABO blood groups and nail patella syndrome. They are linked. They are on one chromosome. Okay, that type of information. Then you go for gene mapping, and genotyping is being deferred. But now, because of whole exome, whole genome sequencing, what we call reverse genetics, directly they go to, without knowing any disease, they straight away. Suppose in a family, they found a disease is being segregating in the family. Several affected members are there and several unaffected members are there. Then we go straight away in the whole family. We take whole exome genome and then do family within family association, linkage disequilibrium analysis, linkage analysis, and we can point out what is the gene, region of the gene, region of the chromosome that is causing the disease. And then in silico prediction models, animal models, all this, we can understand what is the gene responsible. This type of, technological transformations. Now, DNA-based techniques, simple techniques. Even now, these techniques can help us in rare skin diseases like situations, Mendelian diseases. Here, there are, all over our genome, there are restriction enzymes, We have what you call restriction enzymes. These restriction enzymes recognize a site. Suppose in this sequence pattern, CCTGAGG, this particular MST2 restriction enzyme, it recognizes A. Okay. This is found to be useful for sickle cell anemia detection. Only one restriction enzyme, you digest it and you get sickle cell anemia diagnosis, genotypes. Okay, such type of simplicity. Now, another very Nobel Prize winning PCR, this particular technology, Psychia, he got Nobel Prize. What this technology it says do, suppose you have a fragment of DNA. Let us suppose 100 base pairs fragment of DNA is there in your lab, in your test tube. What this technology do is it multiplies these copies. It makes similar copies of 2 copies will become 68 billion copies. 2 copies will make 68 billion copies. This is an exponential amplification. This amplified product is useful for sequencing, okay? Then after PCR, we straight away go to sequencing. There are sequencers, Sanger sequencing, another Nobel Prize winning technology. This Sanger sequencing has been used for whole genome project before the next generation sequencing platforms have come. This particular sequencing, Sanger sequencing is still gold standard, okay? Even if you do other technologies, validation is by this DNA sequencing. It is highly automated, a lot of chemistry inside. We are not bothered about chemistry. You have an instrument, you have a quality control, and you run experiments and you will become expert. And now when you give that PCR amplified product here, you get this nucleotide sequences. Okay, this is the sequence what we got, and here we found a mutation. Reference gene you have C, and in the patient you have T. Okay, such type of DNA sequencer. This is a very, very useful, simple technology, and these two technologies enough for us in Mendelian genetic diseases. Genodermatologists, there are dermatologists who are interested, and several, they have tried to understand the classification and also genes responsible for different phenotypes. And all these, in fact, classification, cognification is one major identifier, and palmoplantar keratosis and acantholytic DOC. And there are, among them, there are non-syndromic and syndromic. Non-syndromic are mostly Mendelian, okay? Whenever I talk about whatever examples I have, I will be talking, they are Mendelian and rare skin diseases, and they are monogenetic, okay? But here you have a panel, skin disease, the companies will come to you with 80 gene panels, okay? And they can put any disease of skin, they put this panel, and they will try to tell you this is what the gene responsible for your disease. But that much paneling is not required when we are dealing with rare genetic diseases. Rare skin diseases definition, rare diseases are defined as orphan diseases, as drug companies not interested in developing drugs. This is a very interesting scenario here. No drug company will come to develop a drug for rare skin diseases because of commercial interest. So this is called orphan disease. But different countries has defined these rare diseases differently. A disease is considered as rare in USA. As per Orphan Drug Act 1983, any disease with less than 2 lakh sufferers in the country, they considered it as orphan disease. And in Europe, 1 in 2,000. And in those countries, the systematic documentation, registries, that is very fantastic. There they can estimate how many incidents of these diseases. But in India, unfortunately, we don't have proper records. Now, people try to list, how many skin diseases genes are there, how many skin diseases are there, how many of them are rare diseases. In fact, GARD, Genetic and Rare Diseases Platform, that is Information Center, out of OMIM, Online Mendelian Inheritance in Man, listed 1,502 skin diseases in 2021. And GARD listed 573 rare skin diseases as of 2021. And Leach and Moses, Online Genodermatology Database, they created a database online. One can visit that site, and slowly, diseases are being added there. Like that, this Lemke, he opined that 80% of rare genetic skin diseases are likely to be monogenetic, estimated to be around 450. Approaches in genetic testing for rare skin diseases, I took example of Malde Melade. What we do when a clinician refers a case, we do extensive background research, published articles, databases. We understand etiology, epidemiology, path biology, molecular basis, and we look into gene annotations. Is it monogenetic, genetic, allelic? What is it? And then we target hotspot genomic regions, and for sequencing, it reduces tremendously the cost. And then when we get a mutation, we validate it in a family for segregation. Malde Melade was first described in 1826 by Lucas Stully, and he said there is abnormal imperfection of integumental imperfection at birth. This particular island, Meleda, now it is called Maldives, is present in Croatia. Disease is known to exist over 50 years in that island. 11 cases out of 3 families without any signs of contagiousness. This man, he has studied 11 cases and he said no signs of contagious. He was talking about genetics, inheritance. In fact, the island is a settlement of lepers and plague. Subsequently, the disease has been reported in at least 19 countries. Okay? Characteristics: classical diffused progressive transgradient hyperkeratosis of the palms and soles, hyperhidrosis, and minor symptoms like erythema, elbows and knees affected hyperkeratosis, finger and nail abnormalities. Maldives, Melody, the problem is of incidence. In the world, they have 1 in 1 lakh population. It was the estimate. And more than 100 cases has been reported in the world. India, ours is the second case being reported. First case was earlier reported by Japanese collaborators with Indian collaborators. And this is our second case. Estimates, as you know, it is not available. But what we did is that we went to Genomics databases. We have 1,000 human genome project for European populations. And we have South Asian Genomes and Exome Project stage of 1,213 for South Asian populations, where Indian whole exome samples are also there. When we looked at the mutant allele, because this is autosomal recessive, the heterozygotes are normal. When I'm talking about Human Genome Project individuals sampled for whole genome, they're all normals. And in the world, the mutant allele frequency is 0.0002. In India, it is 0.001. That means lot of cases in this country, they must be there, okay? Somewhere the documentation or registry or a diagnosis, there is some problem which we would like to address. MDM is autosomal recessive heritable genetic disorders. The gene responsible for this particular disease is called ARS gene, component B. This gene is, in fact, constitutional gene, constitutional. It is involved in DNA replication, a critical step in cell division. Each cell has to divide, and in that constitutional function, like cell division, this gene takes part. This gene is producing, it is encoded by this SLUT protein, okay? And this protein takes part in the cell division processes. But what implicates this particular, this gene produces several proteins, isomorphs of the SLUT1. But this particular protein, which we are interested, SLUT1, is implicated in maintaining the physiological and structural integrity of the keratinocyte layers of the skin. This particular protein is responsible for this hyperkeratosis. Okay, this is the function. These function studies are done by several other animal models. They have understand protein, they isolate protein and look for cDNA and map the gene, all this process. But essentially, the gene is mapped on chromosome 8, 24.3, by whole genome marker, the reverse genetic process in large families of MDM. In Croatia and Turkey, they have large families and they mapped the gene by whole genome mapping. Okay, the length of the gene is 1,457. It's a small 3 exons. The genes have 3 exons, and the exons cover 524. What we can do here is we can amplify whole the region, all the nucleotides we have amplified by sequencing. And up to now, 17 different mutations is associated with MDM in several other countries. The patient here is a 30-year-old male MDM patient referred by Dr. Vadbu Anandakumar. And you see the hypertrophy of palms and hand soles, affected. And the clinical symptoms, characteristics, characteristic of this particular disease. But unfortunately, this person, at the age of 38, he has been diagnosed. Otherwise, disease symptoms are from birth. He told us, the patient tells us that his parents told him from childhood he has got this problem. And he has visited several treatments, processes, and also allopathic, non-allopathic. He went for Ayurveda, but only they all aggravated the disease. Okay, but ultimately, Dr. Ananda Kumar could refer to us. He could diagnose this, and he said he's suspecting Malde Melade. Then we looked into even he suggested the SLRP gene, and we searched all the records, and we found disease may be. We designed our primers, we sequenced, and what we got here is the father had died, no symptoms of the disease, and the mother is still alive, and this person is the patient. He got married, and his younger brother. And we found a homozygous mutation in SLRP gene. C has been replaced by T, homozygous. Mother, we have heterozygous. Brother, we had heterozygous. And wife is normal. In fact, this man, when he came to us, and he in fact came to, in terms of genetics and all that, his basic problem is, sir, I got married. Whether I can produce children. I don't want to produce my children with the disease. I told him, okay, wait, let us see the genetics. I will let you know. Then when I understood this, what is happening here, I told him, yes, your wife is normal. You can go for the children. But remember, children will be all heterozygotes. Okay. Even brother we have counseled. He is willing to, before marriage, it may not be possible for him to ask the wife's tester, but he says after marriage, he definitely, when she's pregnant, he will go for testing. So this type of counseling, has helped the patient family. And in fact, family is in touch with us. This man ranks me up a few days ago, and he said he is blessed with a son. Sir, he has no disease, no symptoms of his hands and feet are normal. Very good. I said, congratulations. He's so happy. This mutation, as I told you, is a nonsense mutation. Okay. The protein truncation after this mutation, stop codon. First time detected in India. This mutation is detected first time in India. Also reported as a founder mutation, Croatia, Turkey, and Korea. They also got this mutation in those countries, and they looked at the similarities between, haplotype analysis. They said only one mutation, that is a founder mutation. Origin is Croatia, and this is now present in Turkey and Korea. Now, research question we are asking is whether Indian mutation is same as examined by haplotype analysis. We want to again do haplotype analysis and understand the origin of this disease. As you know, this country, ancient migrations, a lot of migrations from Europe, okay? And in fact, from here, they went to Europe and came back. A lot of documentation on genome diversity. But we are sure that this mutation also must be same. Let us see. And earlier tested MDM patient has a mutation. It is IVS interlinear sequence 1+Y with a frame shift mutation. And what we observed all over the world, when you look into clinical expression and the mutation, clinical expression is ubiquitous. Wherever, wherever the gene is there, the mutation is there, and clinical expression, there is no heterogeneity. They are ubiquitous. Even people thought, based on the age you age more, maybe it is severe symptoms, but the basic characteristics are diffused, progressive, transgression, hyperkeratosis is always there. Okay? And in fact, why we are so confident with the family advising them, you can go with his child, because this particular epidemiological work, looking at all ethnic populations and looking at the mutation, looking at the clinical, we are confident this is the causative gene. Okay, this type of confidence comes based on several, research, of course, is important. And patient care, you see, chronic disfigured, lesions, incurable. They cannot lead a normal social life. Stress, anxiety, or depression, constant medical support, care throughout lifetime. Our patient was in a Jilla Parishad office, okay, but he's able to write, but he covers soul, he's careful not to expose his hands and feet. He felt earlier he was depressed, but as he learned the knowledge, okay, and we taught him a lot of genomics. Some type of, you see, they understand, no two individuals are alike. Everybody is having abnormal genes. Constant medical support is required here. It is known that these skin diseases are by birth. Early detection is always better in treatment and management. Family counseling, as I showed you, help the patient. Therapy. Now, here the situation is a scenario. No standardized treatment. Topical keratolytic agents are being given. Aromatic retinoids helps the patient in producing improvement in MDM, but long-term use has been found to be toxic in terms of contraception and liver toxicity. That's where we are talking about precision medicine. Translational medicine, this particular gene responsible for this disease, expression is regulated by retinoic acid, epidermal growth factors, and interferon gamma. Novel approaches, this is being followed. Even USA is putting a lot of money in genetic engineering, protein replacement, antisense epidermal growth factors, treatment, etc. Even recent papers, 2019, they said recent advances, role of epidermal growth factor receptors in psoriasis, and also protein replacement in skin diseases like congenital ichthyosis. So a lot of activities going on. And one particular person in treatment of disorder, he recognized epidermal growth factors, receptor inhibitors, is a good candidate. He prepared a lot of permutation combinations of these receptors, and he filed in a patent application. Okay, this type of enthusiasm, this type of discoveries, okay, can happen not in India. Future perspectives, if you look, USA created special institution funds for translational research. UK, 1 lakh genome sequencing project for cancers and rare disease institute, initiated in 2018 and completed in 2019. Several diseases of unknown etiology benefited by novel gene discoveries. Patient groups for specific diseases help networking, sharing information. In India, genomics is in clinical practice is yet to take off. Maybe for rare diseases, there could be a beginning. Genome Foundation is always willing to collaborate with dermatologists in helping patients with genomics. Now, what we are thinking in terms of future medical practice, what they claim precision medicine will be like this. You have all the information, laboratory tests, scans, and X-rays, microscopy, And then you have whole genome, exome, epigenome, transcriptome, proteome, microbiome, even gut microbiota. Okay, all this you put into a computer and processing, diagnosis, genes, pathways, drug-drug interaction, and treatment. This is what is assumed to be it is anticipation and going to be a reality in future. And whole genomics is being a big way in these efforts. And already countries are, in fact, cancer, lot of advances. Okay, but now rare skin diseases with simple technology we can help. Dr. Subha R. Fatke, he is a hematologist. She uses genetics like left and right in hematology. She also teaches DM courses in human genetics in SGPGI, Lucknow. This is the only place where DM in human genetics is given. Her depiction, soul search, all next generation, ATGC, whole genome, 3 billion base pairs is there. And, is it like a soul search? That's what, a little bit of skepticism must be there. It is a reality. Reality is this. Any clinician, they think, genetics may not be of some help. Like in whole genome, I don't know. But this is what, reality may be a skepticism. But what is required is research-driven hypothesis. If you have research-driven hypothesis, whether it is complex disease, monogenetic disease, whatever it is, if it is research-based, you have a clarity. Instead of what is happening now is technology-driven hypothesis. Technology, companies are putting technology on your table, okay? This, even to choose which technology, that should be, you should be able to define that. Okay? Thank you all for your patient listening. I hope I am within time. I am open for any discussions. Yeah, thank you.
Any questions from the audience? Please unmute yourself or raise your hand. Does anyone have a question?
This is Dr. I.S. Reddy.
Yes, sir.
I have a question for Professor Dr. Raghavendra Rao. As a clinician practicing dermatologist, we see quite a lot of cases of T-cell lymphomas, especially a particular type of lymphoma called mycosis fungoides. In this particular thing, one of the important investigation recommended for this particular disease is DNA studies, especially T-cell receptor gene analysis, where we are supposed to see whether there is any monoclonal proliferation of the abnormal T cells. Is there any facility in our institution to do this particular investigation?
Yes, we do handle tissues. Okay, so if it is tumor tissue or blood tissue, whatever it is, we can handle.
Usually we do biopsies.
Biopsies, yeah. Biopsies, if you can send us, we will look into whatever, you want a whole, in fact, what this tissue, they go for transcriptome, okay, in terms of mRNA, did cDNA conversion and go for whole genome applications, okay. We can look into it, but there are several companies who are offering you a customized approach, already they're customized. Those things we have to look into. And we definitely, what we are going to help you is in terms of understanding, whatever you may say, technology. We have Dr. Amitabh dealing with next generation sequencing. We have Professor Vishnupriya, who dealt a lot of this type of cancer, tumors. And what we can guide, you, even a company when it comes to you, I want to do this, we will be able to put you in a proper perspective. Okay. And companies, they are doing a lot of, I don't know about my other colleagues, maybe some companies, but I do good regard for them. They have come forward with technologies. But only thing, they are, we need to have research backup, In USA, Texas Medical Sciences School, when I visited there, each department has got, Genome Foundation type of support. Research staff for one department. Such type of integration, interaction, across the table discussion. So these things we can help problems at all, whether it is technology, simple technology, complicated technology. NGS is one thing, transcriptome is another thing. mRNA sequencing is an expression profile. When you look at tissues, we are looking at expression profile. Okay, all these things we can help you. No problems.
Yeah, thank you very much.
Good evening, sir. I'm Dr. Sharad Chandra from Mahbub Nagar. I'm also a practicing dermatologist. Yes. I happen to come across a few rare skin diseases. Yes. One of which I have sent for gene mutation analysis.
Okay.
It's a very superficial form of epidermolysis bullosa.
Oh, okay.
It's also called McGrath syndrome.
Okay.
So the mutation analysis I sent to another center, but which did not yield the expected results. But the phenotype, as described in there are only 13 cases in the world.
Yes.
So can we do a mutation analysis here in our center?
Yes. See what it must have gone, there. EB epidermis bullosa is well documented phenotype. Okay.
Yeah.
And it is a very rare skin disease. Okay.
Yeah.
Now the question of when you went to for a center, they come with a panel, NGS. They immediately tell you, I will do NGS, next generation sequencing. Okay.
Yeah.
And because technology is such complicated technology, and you have to ask him what is the depth you are going to give. They will say 20x.
Yeah.
100x coverage. They will say 100x coverage.
Yeah.
But nobody asks what is the depth for each gene, each variant. And if you really do this type of NGS for single mutation detection, and it is not a multi-gene involvement, there may be different genes, EB is about 10 to 15 genes will be there.
Yes. This gene is a plecophilin gene 1, sir.
Yeah, must be there. And what this company will do? They want to targeted sequencing of these genes because it is very costly.
Okay.
It's customized. They will put into whole exome and they will give you less 20x because you will not be able to pay for whole exome sequencing what they charge,
Yes.
And 100x, when demand for 100x, their charges go up 40,000, 50,000. But in
we paid 24,000, sir.
Yes, that is 20x. They must have given you 20x. They will miss lot of variants.
Okay.
And they will give a negative report. When I ask them, what is the clinical significance, they say, sir, we are not giving false positive reports. What about false negative reports?
Yes.
They say, sir, 1% is permanent. No, 1% in a diagnosis, a family, okay, if the same thing, if I could have done by this NGS platforms, I could have missed the mutation, such mutation, the family will be suffering. Well, one family is at stake, families. You cannot negotiate for quality. You have to go for 100x depth. I think Dr. Amitabh will be covering all these issues. But as far as your case, epidermal bullosa, we will do it very economically. We will look into the literature. We will design primers. We will sequence, and we will get back to you what is required.
Okay. Sure, sir. We'll get to you.
Right, dermatologist should come together and registry. We need to prepare a registry, for these rare diseases. On the other day, we have one patient, with 2 girls with ichthyosis, lamellar ichthyosis. From birth, they are suffering, both are female. The family is all depression. Okay. Dr. Waggu only sent this sample, these patients, and we are looking into it. Doctor, what is required, among dermatologists, you should have a network and start, documenting. Okay. We will help you. How to make a registry, how to make all this will help you.
So can we send the blood sample of this patient to Genome Foundation, sir?
Usually we require patient. We would like to interact with patient. Okay.
Okay.
Ultimately, genetic counseling, whatever. You see, in my whatever I interact with patients, I only explain to them genetics.
Yeah.
I only explain to them. They are becoming very knowledgeable. They are becoming very positive. Their whole mindset changes. So we insist, instead of sending sample, please kindly send the patients to us. And we have to explain to them. We have to teach them what is EB, what is genetics. Everybody is having some abnormality. Okay?
Here I may intervene here for a minute. Inviting patients, asking patients to come is only for genetic counseling, free of cost, and understanding the entire symptoms and all that, so that our research staff, voluntary research staff, will only try to help the patient, first of all, to have management of the disease, and also, if possible, they will try to give with the knowledge available in all range of genetic testing available. Incidentally, for example, CDFD, Center for DNA Fingerprinting Diagnostics, have signed MOU with us, and one of the MOU condition is, if it is a rare genetic disease, they will do the diagnosis free of cost, because they want to list out all the genetic diseases in India, being one of the research organization, a premier organization in India. So the idea of calling the patient is only to do free genetic counseling and try to see that if it is really a rare genetic disease, we'll try to help him in getting the things identified free of cost. And of course, if we do our own testing, that means DNA sequencing through Sanger sequencing, they will charge a little. That too, Professor Raghavendra will be available all the time to support the patient. Thank you. Anybody else?
Dr. Gandhi, Dr. Kumar here.
Yes, good evening. Please tell me.
Yeah, I have seen a large number of epidermolysis bullosa in Asra Hospital. This is the sister hospital of Ovaisi Hospital.
Yes.
So that is all particularly Muslim population. I might have collected more than 60 cases in that hospital, but nothing was done. So we knew nothing about genomics at that time. So I have retired now already 5 years back. What I suggest is one of these youngsters, like somebody here, so he can collect all the epidermolysis bullosa, bullous disorders, one, keratolytic disorders like Vangu, Anand Kumar, somebody, and some photosensitivity disorders, somebody else, some Gandhi, and some other disorders from Usmania. Like that, if they collect, there's a nodal point, then all of them will come to you. Then it will be, you can collect the data and then easy to publish, and then the group, we collect data also. So then also we need somebody to have a patient association, somebody to guide them, because each clinician cannot give a lot of time for counseling them. So whether to have more children or not, what is the probability, these are all things really a clinician has no time, they just pass it off. So most of them, so there are clinicians who are seeing 100 patients per day. In Warangal, somebody sees 400 patients per day. I don't think they have time. So this needs time. I think this audience, there are good people here. I can see people like Rajalashmi, Vangu Anand Kumar, I.S. Reddy, so many others there, Diksha. I think people can collect cases from, say, somebody in Andhra, somebody in Telangana, then a focal point. So one person can take one group of disorders, then there is a point, starting point that should be the idea, I think. My suggestion, I mean.
Yeah, thank you, Dr. Kumar. I think it's an excellent suggestion. In fact, the purpose of this seminar is a maiden seminar. We thought that clinicians do not find enough time.
Yes.
Because they are busy working with the patients and giving them relief. But we scientists, especially Genome Foundation, voluntary scientists, scientists who came to Genome Foundation voluntarily, like I myself, I'm a volunteering. Lalji was volunteering. So voluntary scientists who are having societal consciousness and national consciousness would like to spend time, and Professor V.R. Rao will be more than happy. Okay. Professor V.R. Rao will be more than happy to meet some of these patients, and we can perhaps create a separate cell with the type of indications you are giving about so many cases. We can create a separate cell in dermatology itself. And Srilata is here, and Pariplavi. Pariplavi, your role is going to be coordinating with all these doctors and patients, especially Dr. Kumar. He has a lot of information, and he told a few names of Dr. I.S. Reddy and a few others. Perhaps Dr. Kumar can be a nodal point from whom you can try to have this kind of contacts. And Professor Raghavendra will act as a nodal point, and you can act as a conduit to him and see that this initiative he translates into some action. Translates action. But Dr. I.S. Reddy also is very keen about doing some kind of work, research work, and this is a good platform we have created. I'm happy that people are coming forward. Professor Rao, you want to say final word, Professor Rao?
Sir. Yes, what we require, as Dr. Kumar is suggesting,
yes,
among dermatologists, they can, take care of, suppose EB is taken by one group, and another group is taking care by other diseases. Like that, among them there could be a group, and through them we collect information and start making registries. Registries we have to make, and wherever possible, we have to interact with patients. That ultimately the purpose is okay, I'm making registers, but we'll also make it a point, a little bit of extent work, talking to patients, trying to talk to them, understand them. Okay, this is one aspect. Other aspect is patient groups. We need to have patient groups. Suppose Malde Melada, I am sure in Telangana there must be several, and clinical symptoms, among the pediatric dermatologists. We need to have a network, share information. What are the symptoms we have to look at, and then, if they can point out this may be that case, then early detection is important. So dermatologists, we can make them groups, we can interact with them more, okay? And it is not the end of one meeting. As you said, we have Dr. Pari, Dr. Supriya, Dr. Srilata, okay? And Amitabh is also there. So we can continuous activity, and ultimately patient is the center of our activity.
Yeah, but the approach towards having this network, ultimately, I want because I'm a doer. I want to know from these interactions what ultimately is the approach and methodology to resolve and help the patients.
Right. Right, Dr. You're right.
Yeah, this is. I think you should come up with some practical suggestions how to go about. Dr. Pari, I think some of these inputs we have to take with the name, and we'll be able to identify their email IDs and perhaps we can form a small WhatsApp group, Dermatology and Genomics of Genome Foundation. So if it is worthwhile, if some of you agree that this can create a worthwhile group to go forward, we can always do it. Genome Foundation, Dermatogenomics group, something like that. Dr. Kumar, can you just comment on this?
Yeah, I think we should start like that because there is somebody is interested in something, then they can channelize it. For example, I was studying xeroderma pigmentosum in Alin Institute. When I sent the letter to all the other doctors, I have collected 35 cases in Delhi. 35 cases. I did not even publish because I had to leave due to some personal problems. That study went just like that. And secondly, that was about prognosticating xeroderma pigmentosum based on sunburn cells, etc. So which I was doing. If somebody is interested, all other people, because this is academic forum, so everybody says somebody is interested, so let me pass it on to him, he will do something. So that will be usually when rare diseases, everybody wants to know either something is being done there, we'll send the cases there. So Genome Foundation is doing there. So everybody will say after some time, go there, something will be done. If there is an answer, you will find it there. So that will be a focal point. So there are a number of youngsters also interested academicians in this group. So I will try to find out this Dr. Sudharani, Rajalashmi, Dr. Chandravati, I.S. Reddy. I think my friends here, I think they will help in identifying some groups. So we will do it.
Yes, the doctors at Osmania and Nilofer said also.
Can I talk, sir? This is Dr. Bhanuja Rani, sir. I'm myself, I did my biochemistry also. So I'm doing now SLE as a genetics in SLE. As a cystatin C, sir, I have done that gene analysis. In that, I found there is a renal involvement with cystatin C. Lot of that gene is specific wherever involvement of renal problem, we're having CST3 gene. And one more project I have done through MDRU is vitiligo. I have started recently. I have to analyze that. In vitiligo, the genes in vitiligo, I want to analysis that. So these I'm doing through MDRU of Gandhi. Recently, I got promoted to Usmania, so my project is there. But why, can we not only to genetic diseases, can't we go to the common diseases? And what till now you are insisting more on genetic diseases, and that is my opinion, sir. Why not we can come to common diseases also? The genes, anything. If you see SLE, renal involvement, SLE with cardiac involvement, what are the genes there? What is the gene specificity, and why, if this gene analysis was done early in SLE patient, we can predict whether they are going towards cardiac complications or they are going towards the renal complications. This is my thought, sir. I don't
absolutely. Dr. Bhanujarani, absolutely you are right. We are not limited to rare diseases only. We are also taking other aspects also.
Yes, sir. So since 4 years, I'm doing this research, sir, in Gandhi, MDR, through MDR.
Good. Professor Rao, you can also react, saying that.
Where do you do genetics, madam?
Sir, their MDRU is there, sir. They are providing us the material. Yes, sir.
Genetic testing, where
we are doing that. The scientists are there, sir. Dr. Madhavi and Dinesh are there.
Okay, you have a setup, good setup there.
Yes, small setup.
You are rightly said, madam. SLE is a big problem. It is increasing. And what our KIMS, we had recently a discussion with rheumatologists there, and there is a possibility, actually, what is important, is like this. If all any intervention, therapeutic intervention comes anywhere, okay,
Sure.
But Indian patients, because of genomic diversity, we do have patients, clinical and genetic correlation, expressions. Whatever repository we have, nowhere in the world we have that. Unfortunately, our patients are not documented. It is you are doing a project, madam, okay? Like that, several people are doing projects, okay? There is no ultimately, whatever, what you call it, a huge investment from central government or any governments, like 1,000 genomes. Who thought about 1 lakh genomes, madam, whole genomes? Immediately, UK, what they did, ultimately what they do, the data will be sold to private companies.
Okay.
That type of, commercialization is also there. But we are no poor researchers. We want to do something, and but our diversity sample, patient diversity, it is very, very valuable for discoveries. I'm sure you can find cardiomyopathies we have shown out of 60%, genes, novel genes. Only 30% candidate genes are there. Hypertrophic cardiomyopathy, RCM, DCM. Candidate genes, we got only 30 families. Another 60 families, novel genes. Is a discovery. And we are going for zebrafish and all that, animal model, all that. So there has to be translation, only one institute for the whole country, Translational Research Institute in some Delhi, Tisha, something like that. Amitabh, what is that institute?
THSTI, Translational Science Institute. THSTI,
Faridabad. Of this huge country, one national institute. And there's national institute, whoever is the project they are conducting, they're all small projects.
Yes, sir.
Yes, madam, whatever help you want.
We can approach, we can take this SLE also, we can do a good study. Yes, sir. We can continue the study. That is the mean to say, sir. I mean to say. Yes, sir. Because for me, it is, I have done only for 50 cases only, as SLE is a rare disease. But all the dermatologists, if they are willing to come, we can get more.
Right.
Okay,
right,
sir.
Yes.
Madam, Dr. Pari,
Yes,
Dr. Vakku Anand Kumar is with us. He is the man, and
who
Dr.
sent
Sai
you
Kumar,
this.
he is very active in sending. If you want to interact, you want to say anything.
Yes. Dr. Anand Kumar, would you like to say?
Dr. Vakku Anand Kumar.
Share your thoughts?
Or Sai Kumar is there. I saw Vakku Anand Kumar somewhere.
So he may have left or.
Yes, he's probably left.
Okay,
Shall we move on, sir?
Yeah, please.
Yeah. So this was a very interesting and fruitful session that we had till now. Dr. V.R. Rao with a very simple and lucid presentation with wonderful slides. He spoke of the paradigm shifts in our understanding of both the diseases, the processes, the procedures, and the technology that are going on, and how we need to learn and keep alert and keep updating ourselves to be able to adopt new technologies and the new concepts. He spoke of why skin diseases are easier to study, explaining how 80% of this genetic skin diseases are monogenic and with 100% penetrance, and therefore very ideal for genetic studies and very ideal also for prevention, for counseling, for the development of translation medicine procedures and hypothesis. And not just that, also for anthropological studies and phylogenetic studies. We learned about SAGE, the South Asian genome exomes, and he spoke of how surveys and documentation are important for prevention and cure, and the enormous burden of genetic disease that our country everywhere, but especially a country, a developing country like ours, is facing tremendously, and the psychological burden of that. He spoke of the future of medicine, how it is precision medicine. And most interestingly, he spoke of how we should have research-driven hypothesis instead of technology-driven hypothesis. So, and with a wonderful cartoon that spoke of soul search. So a soul search-driven hypothesis instead of a machine search, machine usage-driven hypothesis. Thank you, sir, for that wonderful insights that we all got this evening, and with very productive offshoots, which have led to such fruitful discussions to plan the way forward. So there's plenty of work for every person here to carry it forward. As Sir said, this is a volunteer-driven organization with those motives. So we entreat all of the attendees here to participate actively in this. So now we move on to Dr. Amitabh Biswas, who will be explaining to us how the next generation sequencing technologies are so useful in clinical dermatology, in the screening and diagnostics. So welcome, Dr. Amitabh.
Yeah, before Amitabh starts his
slides
presentation, I have one small request to all the people who are on, who are now listening. Dr. S. Kumar, Dr. S. Reddy, Dr. Bhanuja Rani, and another few.
Dr. Chandravati.
Chandravati, yeah. They have given their viewpoints, and I invite, first of all, I thank and I invite all those people who are interested to network with the Genome Foundation for the common cause of helping the people in a simple way through genetic means. How best they can have their genetic burden reduced through counseling, through simple testing and all these things. So, because genetic basis for their problems should be first of all made to understood, and Professor Raghavendra will be always happy to do that counseling work, and remaining all our scientists and medical scientists will be able to help these people. I invite you to this forum, and you can call Srilata, who is one point contact for now, and Dr. Pariplavi. You can call them and then try to suggest us the ways and means to network together. Thank you all.
Yes. Okay, sir. Thank you.
Yeah. Amitabh.
Yes, sir.
Yeah.
So can I share my screen?
Yes.
Can you see my screen?
Yes, we can see your slides. You'll have to speak a little louder, Dr. Amitabh.
Okay. So thank you, ma'am, for my introduction, and thank you, Professor Rao, for inviting me here in this webinar. So my topic for today is next generation sequencing and its application in clinical diagnostic and dermatology. So I will start with one of the announcement which was made in June 26, 2000 by British Prime Minister Tony Blair and American President Bill Clinton about the human genome, which has been roughly mapped. So that was the major turning point in the human genetics area. So in this today talk, in 20 to 30 minutes, I will be covering like what is the overview of genome and genome sequencing, what is the basics of next generation sequencing and technology, and what are the approaches we can take on, and we'll take some of the successful examples of this NGS in dermatology. So if, as Professor Rao already has covered, what are the different kinds of intrigues are there in the genome. So I will say that there is a total genome of 3.2 to 3.3 gigabases are there. So human genome is comprising of 3.2 mega base pairs, 3,200 mega base pair. If you divide into two parts, there are genes and gene-related sequences that is around 1200 MB, and there are intergenic region that is around 2000 MB. So if we see the genes and genes-related sequencing, only 1.5% of the whole genome contributes to the coding part, which is very important to study on, which we cover in the whole exome sequencing. And the related sequences which are with the genes related sequences are like pseudogenes, gene fragments, intron, and UTRs. Now, in the other side of the genome, we have an intergenic DNA, which is around 2000 MB, in which interspread repeats and other intergenic regions are there. Like interspread repeats like lines and signs are there, long interspread nuclear elements are there, short interspread nuclear elements there, LTR regions are there, transposons are there. So these contribute a majority part of the genome. Now,
your sound has gone, Dr. Amitav.
if you want to understand,
your voice is breaking, Dr. Amitav.
hello.
Yeah, you can.
So host has muted me also.
Oh.
So for the linguistic term of analogy, if you see, we have a 26 letters, and we can make words from by combination of those letters, and then we make sentence, which makes sense. So like that, genomic also work. We have a genetic code, will make a genes, and all genes working together will make a genome. Now, if we go to next slide, so we want to understand the mutation. I have taken an analogy of linguistic only. So the original sentence is the fat cat ate the rat. Now each codon, as the Professor Rao has told, there is a 3 nucleotide will make a codon. So I have taken a 3 letters for a word to be act as a codon. So codon 1 to 6 is there. Now if there is a change in a single letter, like R is changed to B, thus the meaning of the line changes. The fat cat ate the bat, but the sentence is maintained. So this is a kind of a substitution mutation. Now, if we want to see the deletion mutation, now this T is removed from here, but our open frame shift will read 3 as a codon. So the next letter will shift and it will make a nonsense out of it. So we are not making any sense of this sentence. The fat say ate at her ate. Similarly, if there is an insertion is there, then also it will not give any meaning to the sequence. So these kinds of mutations are there in the genomics. Now, how to do the genome sequencing? Now we know that there is a 3.3 giga base pairs there. If we start reading those giga base pair like a 6 letter per second, then will take 9.5 years to read out loud without stopping. So is sequencing is as simple as reading a book? The answer is no. So how do we do the genome sequencing? Now with the Human Genome Project, we got the reference newspaper. Like we take a newspaper analogy. If a newspaper is a whole genome, now we have several copies of newspaper. We shred that newspaper into the pieces and then assemble to the original or reference newspaper to get to know what article is where it is. So similar kind of thing is done in NGS also. Now, if you see the DNA sequencing timeline, starting with the DNA structure in 1953, we have got the Sanger sequencing in 1977, then polymerase chain reaction in 1983. Then the Human Genome Project was announced in 1990, which was completed in year 2003. So this was the era of the first generation sequencing. After the publishing of Human Genome Project, the next sequencing generation boomed. There are now we can do 100 of genome in 2 days. The first genome to done is required 13 years. Now we can do it in 2 days, 100 of genomes. So this is the technology boom we have in the next generation sequencing. So if we talk about the first generation sequencing, that is Sanger sequencing, which was developed in 1900s and published in 1977. This man has done this work, Frederick Sanger. He got a Nobel Prize for this work. Now, if you understand the Sanger sequencing is a simple sequencing method which require 4 material like primer and DNA template, DNA polymerase, D-deoxynucleotide phosphate with fluorochromes, and normal DNTPs. So if we have a DNA template, we have a primer for the target sequence. Now, if any of the deoxydinucleotide nucleotide attached to the primer, this sequence will terminate. So every time any of the fluorophore deoxy terminator nucleotide will attach, this terminate the reaction, and this will give you a molecule of different sizes with the fluorophore. Then these molecules will go to the capillary gel. On the basis of their sizes, the sequence will get, the molecule will get separated out. After each separation, this will be detected by laser. It will be emit fluorescent, which will be detected by the computer and result in the chromatogram. You can see the different letter has a different chromatogram, and we can read the sequence. So this is the Sanger sequencing. Now, what is the utilization of Sanger sequencing? For the routine sequencing application, we are using Sanger sequencing, and also to validate the results from the NGS, because it's a gold standard sequencing for any kind of results. Now let me talk about the Human Genome Project, which I started with. This Human Genome Project took 13 years and $3 billion to complete. And hundreds of scientists were involved in this project. So they not only sequence the human genome, but they sequence bacterial, yeast, worm, fruit fly, and also number of animals' genome also. So they have not taken from, they have sampled from a single donor. They have taken from the multiple donor and then assemble the sequence. So this sequence was done with the 99.99 accuracy. And they have annotated like 3.7 million human SNP in this Human Genome Project. Now, what is the impact of this Human Genome Project in biology, technology, and in medicine? So in biology, we can now take on with the system biology, how different mechanism, different pathway are interacting, we can study now. And also with the multi-omics, means we can integrate transcriptomics, we can integrate metabolomics, and also the proteomics part within, and also in the evolution part. Now with the big data, now we are generating huge amount of data. So there is a technological boom in computational science also. So we have to manage the big data. So we need a big data science also. So there is a technology-wise impacted also there. In terms of medicine, there is a variation in the medicine. We have developed haplotype, we have developed 1000 genome project, we have done number of GWAS studies are there, genome-wide association studies are there, which are based on this Human Genome Project. Now we'll understand what is next generation sequencing. In general term, next generation sequencing is the million of reaction performed in parallel. So in Sanger sequencing, we are doing only one reaction at a time, but in next generation sequencing, we are doing millions of reactions in a parallel way. So it's the massively parallel sequencing. So if we're doing it in a million of reaction in parallel, so we have a shorter lead length and higher error rate. But nowadays, the technology is improving and they are giving long read length also, and error rate is also decreasing. Now this NGS facility, if you want to use, then you need to prepare your sample. You need to prepare the libraries from the sample. That is one of the drawback. But in Sanger sequencing, you don't need to prepare your sample. You need to do just PCR and do the sequencing. Now there are many platforms are available based on different approaches, like Illumina is doing by sequencing by synthesis, Roche is doing by pyrosequencing, Iron Torrent is doing by pH indicators. So there are different technology with different chemistry. So every platform has a same fundamental that they need to do the thousands of reaction massively, parallelly, clonally. So Roche is doing ePCR, then fluorescence, whereas sequencing by ligation, which is done by solid, is ePCR and fluorescence. Whereas LifeTech or Ion Torrent is using emulsion PCR and followed by what is the change in a pH. And Illumina, which is the major giant in sequencing field, they are doing by clustering, which is sequencing by synthesis, and then fluorescence. Now, if you understand what is a basic workflow from sample to data analysis in next generation workflow, so we have a biological sample, we extract the DNA, we'll make the library, we do amplification, and we do the sequencing. We get the sequencing data, then we'll check the read quality, map the gene with the reference genome, then we'll do the post-mapping controls like removing the duplicates, recalibration, and all the things. After recalibration, we do annotation. Annotation means is the mutation is single nucleotide variation, is the indel is there, any copy number variation is there. Then after annotating the variant, we'll go for the data utilization part. Is it used for druggable target identification or phenotypic heterogeneity, or any we want to identify the causal mutation? Like that, whatever the question is, we utilize the data for that. Now, if we understand what is the basics of next generation sequencing, is the making the libraries. Now, all the platforms are using similar kind of workflow, like first to fragment the DNA. This DNA can be mechanically or shearing by fragmenting the DNA, and then it can be done by enzymatically. Then we have to repair the end of the DNA. Then we attach the detailing, and then we attach the adapter, which is specific to the whatever the platform you are using. If Illumina is using you are using Illumina platform, then you will use a specific adapter for that. Then you will do amplification, and then these library construct will go for the sequencing. Now, if you see that this is the Illumina construct is there, library construct. This is a DNA insert. We have a 2 read sequencing primers are there, so that we can read from this side also and this side also. And we have a dual index, which to identify the sample, because we are pooling many sample in a one tube. So we need as identifier. So this is a barcode which is unique to the samples. And then we have an adapter, which are specific to the Illumina sequencing platform. So we need to attach this adapter. Now we have the raw reads after sequencing. We do the mapping. Mapping means we are aligning to the reference genome. Then we do some local realignment. What are the duplicate? We need to remove that. Do the base quality recalibration. Then this is analysis is ready for the variant discovery. Now we need to discover what are the variants are. SNPs are there, indels are there, all the structural variants are there. After doing this, we have the raw variants are there. Then we'll match with the external data. We want to know what is the frequency or it's a novel variation.
It's 8 o'clock.
So we want to do all these kind of things. We compare with the population structure, known genotype, every kind of things we'll do, and we'll calibrate the variant, and then we do the genotype refinement. After that, this is analysis. These variants are ready for the analysis or what kind of filtering you want. So this is a simplified workflow of the variant, how it is coming out from the machine and how we are getting in the form of. So we have a BCL file, base call files are there, which is converted to FASTQ file, which is also known as demultiplexing. Then these FASTQ file are mapped or aligned with the reference genome. Then we get the sequence aligned map files, which is SAM files. Then these SAM files are compressed to BAM files to reduce the space, which is binary alignment map files. And then this, after getting the BAM file, will call for the variant and we get the VCF files. And then these VCF files are annotated with along with the reference sequence. Now, what is the overview of the processing and filtering of the variant? We have like all 30,000 variant are coming from the exome. We'll check for the zygosity and exclude the known or common variant. If I'm taking this example for the rare variant, or the rare diseases. So we'll exclude the known or common variants which are in DBSNP or 1000 genome, which have a allele frequency of greater than 0.5%. And then we'll predict the functionality based on the multiple bioinformatics softwares are there. We'll predict the whatever the variation is. Then we'll check the prior biological knowledge, what are the candidate genes and pathways, and we'll look for the this variant in the relatives or to check what is the association is there. And this variant when confirmed, we'll check in the unrelated cases also for any kind of association. Then we'll get the causal variant. So I am taking this example for whole exome sequencing, like we have 30,000 call variants per call. We'll do the QC filter, like we check for the depth, we check for the coverage score, we check for the allele frequency. Then we'll get around 2,000 high quality variants. Then we'll do check with the what is the frequency in 1000 genome or European data or in our South Asian data. Then we'll left with the 20 rare disorder specific variant. Then we'll see the what is the functional effect by using the multiple softwares are there to check the functional effect. And then we'll check also the what are the familial variant. If we have the family for that particular disease, then we'll check with around the family in unaffected and affected members. And then we're left with the 2 or 3 candidate genes that will be possibly related to the clinical indications. Then we can go for the further analysis. So these are the bioinformatics prediction software. There are multiple software available in the market. So recent study has shown that the 2 software, mutation tester and MPPS, that is mutation pathogeneity prediction software, has given the maximum 96% pathogenic variant correction and 86% for neutral variant. So they have predicted nearly 96% as correctly. So we'll come to the sequencing approaches. Now we know that there are 3 kinds of approaches are there: whole genome sequencing, whole exome sequencing, and candidate gene or targeted sequencing. So whole genome sequencing means we are sequencing the total genome, that is 3.3 gigabase pair. Whereas whole exome sequencing means we are sequencing only the exons part, which are the coding part. That is around 49 MB, which is around 1.5%, which I said in my previous slide. Whereas candidate gene or targeted gene means we are targeting the specific genes which are related to disease or whatever your question is. So for whole genome sequencing, we say the drawback is that we don't have the lab, we don't have the standardized tools to to predict the functional variant which is present in intergenic region or like that. So this is one of the problem is, and second is it's very cost and computation intensive because you have a big data, you need a specialized person to handle that data, and you are generating a lot of data. So whole genome sequencing is a costly process. If we see the candidate gene, now candidate gene cannot identify a novel gene because you have already selected the genes what you want to sequence. Secondly, there are some companies which are providing the panels for the different diseases, but that panels is either based on the European databases or American databases. There is no Indian database. If we are using those kind of panels without looking at our literature, then we are failing to detect any kind of variants. So whole exome sequencing is the best bet for any kind of clinical diagnostic. So there is a new word also coming up, clinical exome in India perspective. So what is the difference between whole exome and clinical exome? Now whole exome is the coding region across entire genome, means we are sequencing 20,000 plus genes, whatever genes are there, their coding regions are there, we are sequencing all those. Whereas in clinical exome, we are only doing the limited set of the genes, that is 3,000 to 6,000. Which is present in OMIM and HGMD databases, which are the databases which are reporting the genes which are associated with the different genes. So although we are doing these relevant genes from the set of 3,000 to 6,000, but we don't have the advantage of clinical exome. I will say that clinical exome has some advantage because it is cheaper than the whole exome. It is simpler. It has a less data to handle. But what is the advantage of whole exome sequencing? That if there is a chance of reinterpretation of your data, if you are not getting any kind of things which are related to candidate gene, you can tweak to whole number of genes. 20,000 genes are there. So you can go and do some analysis, go back to the analysis and do any kind of reinterpretation. But reinterpretation has very little value in clinical exome sequencing. So there are some technical terms in clinical NGS is there, like what is sequencing depth or depth of coverage. So sequencing depth is the number of times given nucleotide in a genome has been read during the sequencing run. If I use like screen and pointer to show you. Suppose we have fragmenting, this is the gene we want to sequence. We have read fragments like that. We have sequenced like that. We have sequenced small segments of that particular gene. So this is the gene we want to sequence, and these are the fragments which are sequenced by the NGS. So what will be the depth of the sequence will be the how many times that has been covered. Suppose this is a locus we want to see. So there, how many times this has been covered? 1, 2, 3, 4, 5, 6. So 6 times this has been covered. So 6x is the depth of particular locus. And what is the coverage? Coverage means this gene is covered by the sequences or not, that we have to see. So if we combine, we arrange them, we see that this sequence is completely covered. This region is completely covered. So 100% coverage, but depth is 6%, 6x. So you understand what is the meaning of sequence depth and sequence coverage. So if we go about next is variant annotation. Annotation means we are linking the sequence variant with the functional information, whatever available in the literature. Then what is the variant calling? Variant calling is that we are either calling homozygous, heterozygous. This is the identification process in the bioinformatic tool. And reference sequence I have already told. Now there are many reference sequence panel are available for South Asian, for European. So it is good to we should take the reference panel which is more similar to our reference. Like for Indian, we are using South Asian panel as a comparison because they are more closer to us. So what are the clinically grade recommended parameter for clinical NGS? For whole exome sequencing, it is recommended to go for 30x. If you can see this publication in Nature in 2008, which has shown by experiment that after 30x, the number of SNPs called remain the same. And if you see the missing of the discordant or calls, that also remains same after 30x. So that's why it has been conveyed that 30x global coverage for whole genome sequencing is the clinical grade. Whereas if you see in terms of, if you want to do a 30x, how much space we required in the flow cell in terms of Illumina sequencing, like 3.3 GB is the data, is the sequence whole genome, and we want to do it 30x. So expected generation space required is 99 GB, like 100 GB. 100 GB space is required to do average 30x depth for the whole genome sequencing. Whereas for the clinical grade whole exome sequences, it is advisable to the 100x or more. Like 100x and 120x has been done for the clinical grade exome sequencing. So this is the latest paper. They have done the clinical exome sequencing from different companies, and all of them has done with the 100x or greater than 100x. So this suggests that if you want any kind of clinical diagnosis from the exome sequencing, then go for 100x or more than 100x global coverage for the global depth for the sequencing. Now there are the already published guidelines are there for reporting the NGS that has been published by the American Society of Medical Genetics, and they have published many of them. So you can go through those, and we can all variants can be go through those. Whatever the recommendation is there, then we can go for some clinical diagnosis. They have also recommended for incidental findings. Now, what are incidental finding in 57 genes? So if we are looking for a specific disease, and if we find the causal variant, then report it. But they said that if we are doing whole exome sequencing or whole genome sequencing, there are 57 genes which are associated with the 27 serious but treatable disease. If you find any of the mutation in these 57 genes also, then you should also inform clinician about those 57, whatever the variant you find in the 57 genes. So incidental finding is whatever the variant you are finding which are unrelated to disease of interest. So it may be a small number like 1 to 5 might be well described disease associated mutations. So let us come to the dermatology part. I will be discussing with you some of the successful story of dermatology with respect to whole genome, with respect to whole exome sequencing. So making an accurate diagnosis of inherited skin diseases can be challenging. So just these kind of genetic testing is a valuable part. So NGS, with the advent of NGS, this sequencing and finding a new gene is very easy and also gives a significant effect in the patient's care. Now, if we look at a decade of between 2009 to 2019, NGS discovered 166 new inherited skin disease association and to characterize the 63 multiple gene pathology. So from in those 10 years, 166 gene has been newly reported, and approximately 90% of discovery were made using whole exome sequencing. So why I'm stressing about the whole exome sequencing is that because it is making a huge impact in the clinical findings. So whole exome sequencing, whole genome is the ultimate thing, but whole exome sequencing is the surrogate for whole genome sequencing, which can define the clinical diagnosis. Now, if you see the genodermatosis down the lane, we have number of genes which has been identified from 2009 to 2020. Starting with the 2009, the first successful application of whole exome sequencing, where in the Miller syndrome, they have found the DHODH gene to be associated. Now, I have taken this example of Indian family of congenital laminar ichthyosis. This is a rare disease with very rare incidence. And this disease had previously known with the 9 genes with 149 exons. If I want to do it with the Sanger sequencing, I required 149 reaction to be done, at least 149. I mean, it can be more because if exon is bigger than 1000 kb or more than 700 base pair, then I have to prepare for the 2 reactions. So this involves huge cost and also the labor time. So this is the family pedigree. We have both parents are normal, but both their offspring are having the hyperpigmented fish-like scales all over the body. The clinician done the karyotyping, they find a 22 key 12 plus duplication, but they have found in the father also. So this does not give the answer. So they went for the whole exome sequencing. Now, whole exome sequencing identify a novel mutation in TGM1. So TGM1, they found that autosomal, the both parent were heterozygous, whereas the both sibling, both offspring were having the homozygous mutation. So that's why they are getting this kind of scales pattern. So if you see, this is the mutation glycine 218 to serine. They have got this mutation. This is a novel mutation in TGM1. Now, what is the good thing about this NGS is that for the next child, they have done the prenatal testing for this particular mutation. They found that similar kind of hetero, what their parents got is there, and the child born with a normal skin. So first application is this. Second, after a few months of disease, this publication, this was covered in the print media also. Now they have started, the clinicians started with the retinoic acid treatment, and you can see the improvement in from the publication, from what they have started with the sampling part, and you can see they have a hair and they have no alopecia and all the things. So this the child improved within few months of the treatment, as shown in the print media cutting. So this was the first successful case. Another I have case took from the literature is epidermolysis bullosa combinare, EBS simplex. So this is also a rare condition. This condition they have found in a Gujarat family, multiple number of affected patients were there in this family. You can see in the 5 generation of this pedigree, and there were multiple number of affected individuals are there. This large non-consanguineous family was residing in Gujarat. So they have taken around 14 samples, 9 affected and 5 unaffected. And they have done whole exome sequencing for 2 individuals. And they found one mutation in KRT5. And they replicated the mutation in all other family members. And they found that all affected members have this heterozygous condition, whether and other unaffected member doesn't have this mutation. So this confirms that this mutation is the part of causality for EBS. So this is the one other case. Another case of dystrophic epidermolysis bullosa is there. In this one girl having this these kind of blisters in all over the body, and the clinician suggested for the whole exome sequencing. They did the whole exome sequencing, and but there was no mapping for the disease in India, so they did the whole exome sequencing, and they found one mutation of differential deletion in collagen 7A in trios. And you can see that the mutation here, our family, both mother and father are heterozygous, whereas the child was the homozygous for it. So it's a recessive kind of disorder, and it produces the successful story of one of the cases from India, that they have easily identified the gene, what is the causation for that particular disease. Now I have taken another example. This is not from India, this is for psoriasis. But we know that psoriasis is a multifactorial disease. It's not a rare kind of things. Or it's not a monogenic disorder. It's a multiple gene disorder. So in this, they have seen what is the accelerating response to psoriasis patient. They have taken 179 patient and divided into discovery set and also a replication set. Discovery set, they have done the whole exome sequencing in 13 patients. And whatever the results are from the whole exome sequencing, they have replicated in 166 using mass array. So they found that 4 SNPs dominantly or codominantly is associated with the accelerating response, either in the codominant fashion or in dominant fashion. So whenever these markers are there, this response is good as compared to other. So this also is, we can give the example how whole exome sequencing can be used in the treatment as well. So whole exome sequencing is playing a crucial role in diagnostics as well as in treatment. So because doing any kind of genetic analysis, we require a good phenotype also. Because if we have a good phenotype, if we relate with the what kind of analysis we are doing in genetics, will give us the results. So it is very essential to have a good phenotype. Then only we can overlap the result with the phenotype and come to any conclusion. So what are the clinical indication that you should go for a genetic testings when we have a cases with suspicion of genetic etiology with no clear diagnosis, like multiple clinical features that do not suggest specific disorder or with unusual presentation? We have a phenotype with many associated genes, then you should go for a genetic analysis, or clinical diagnosis for which no targeted testing is available. Then we should go for a genetic testing. or complex cases with no clear diagnosis, or previous testing that is negative and only account for the part of the phenotype. So these are the clinical indication that you should go for the whole genome sequencing kind of things, or whole exome sequencing. Now, if we see the what is the diagnostic yield among the different area, we have seen that dermatology has the highest yield, diagnostic yield, like 60%. If you see the 60% yield is there for dermatology. So if we apply the NGS technology to the dermatology field with the clear perspective, with a clear phenotype, then we can get the result in a positive directions. Now, what are the challenges we have? Now, NGS data density, we encounter various reports with the variant of unknown significance. So how to deal with it? Which variant is clinically actionable? What is the evidence we get from the populations? What are the risks of overinterpretation? And you need to carefully select the patient for genome sequencing and genome counseling. It's crucial. Then only we can avoid these kind of challenges in NGS. Now, how to manage the genetic variant? Now, pinpointing a subset or pinpointing a variant in NGS is very difficult. So that's why I have made one slide that phenotype is important. Until and unless we have clear-cut phenotype, we can't interpret the genetic as or correlate with the genetic in a good way. So ACMG and APM guidelines, they have provided how to interpret sequence variant. They have listed out all the things, what type of variant should we go for, which kind of analysis. So we can go through those kind of guidelines and give the report. And what is the receipt of VUS result is given to clinician, that should not be considered. If any company you are giving you a, like it's a variant of unknown significance in this gene or that gene, that should not be considered by a clinician in any kind of decision making. Now, if we talk about the effect size of the genetic variant, if you see this diagram, if effect size is more, their frequency is less, then it's a rare kind of thing. It's directly posing the to the phenotype. Whereas in polygenic disorder, effect size is less, but the frequency is very high. In rare kind of disorder, frequency is less, but the effect size is more. So oligogenic means both are in midway. So this is the diagram which shows clearly how to deal with the rare disease and how to deal with the common disease. Now there is a question of what is the difference between clinical diagnostic versus research sequencing. Now this word is very important word, validated and regulated. Until and unless any kind of diagnostic tool or genetic diagnostic tool is validated and regulated, it can't be used for any kind of clinical things. So that any kind of test you are doing, if it is required, it is regulated and validated by that company, then go for that test. So at last, I will talk about what is the workflow that should be taken through for the NGS-based genetic testing. First is clinical assessment. A good clinical assessment, phenotype assessment, thorough with the patient history, available exam, and what is the necessity of the NGS-based testing. This is done by the clinician, and then they can discuss with the whatever geneticists and whatever they are contacting companies, what kind of panel, if there is a panel is available, if they want to do the panel or whole exome sequencing or whole genome sequencing. If they want to do the trios or only the probands, what is the turnaround time, how patient can afford that test or not. Then we have to do with the variant management. Whatever the variants are there, you need to filter out methodically according to the ACMG guidelines and then annotate it. Then what is the relevant of public variant database? Like if you are using European database or 1000 genome, then what is the reliability of that? Improvement in the clinical guidance. Then you have to look for the incidental findings, which I have already mentioned that you should look for those 57 genes also. Then what are the best practices after that? you need to check for the what is the accuracy and reproducibility of those results. Means if you have done the NGS, you need to check with the Sanger sequencing method. And we need to check what are the pipelines they have used for the bioinformatics. Is it regulated? Is it accepted by the ACMG guidelines or not? Then we'll come with the return of testing. Now, how to convey the result to the patient in the form of genetic counseling or clinical decision making, then you have to do that. So this is the workflow you have to follow to give the, I will say, good and correct clinical diagnosis to the patient. And I will end my presentation here. Any kind of question I will take.
The attendees like to interact with Dr. Amita. Any doubts, clarifications? Thank you, Dr. A.S. Kumar, for your comments.
Thank you, Dr. A.S. Kumar and Diksha, who have circulated the information to many of their colleagues, and many of them have come. Diksha herself was here all the time, and Dr. Kumar has given a lot of suggestions. I'm sure that he will join our group. And I want Dr. Pariplavi to identify the enthusiastic research-oriented doctors whom we identified, who expressed their doubts about the patients and were keen to know more about genetics. We have to identify them.
Yes.
Kadali, Srilata, you also should identify the names of the people.
Yes, sir.
Anyway, we are going to have a small group formed with 5 or 6 contact people who have come forward. And anyone else, Dr. Rajalakshmi wants to speak. Dr. Rajalakshmi, you can go ahead. You have to mute yourself.
Unmute yourself.
Yes. Dr. Rajalakshmi, we are not able to listen to you. She has to set her.
Please unmute yourself, ma'am, and you can cut off your video if you're having an internet problem. Your audio is muted.
Dr. Rajalakshmi, you can speak now.
Now you, yeah.
She's not able to speak.
It's mic problem, sir.
She's having some.
Is there any problem from our side, Karna?
No, sir. Rajalakshmi mic problem, I think.
Mic problem. Okay, okay. It's all right. Yes. Okay.
So,
anyhow, she is with me in Apollo Secunderabad, sir. I think I will interact with her. She is in liaison with all other female dermatologists. They have a group here. We have a very strong female dermatologist group. In fact, they are the majority now. They started when they were in minority. Now they are outnumbering us. I think I'll take their help and then we'll deal.
Thank you very much, Dr. Gandhi, Raghavendra Rao, and Dr. Amitabh. And last but not least, it is but for Kumar. He was the one who informed me about this program.
Yeah, that's why.
I would not have known about it. Thank you very much, Dr. Kumar. And I'm sure we will try to form a group and we will interact. I have a lot of interest in this particular area. I think we should keep in touch. Thank you very much, sir, for giving me the opportunity.
Yeah, thank you all once again. I think if anyone wants to join, Dr. Rajalakshmi, are you able to join now? She's still not able to join. I think Dr. Kumar will help her in joining in the group and then start and continue the dialogue.
Okay.
And I heard a couple of other lady doctors also, Dr. Bhanuja Rani and other lady doctor also. I don't remember her name. Someone spoke. So, Srilata, I think we have record of all that.
Yes, sir. There is a record.
Yeah, we have record. So try to. I think Dr. Pari, your specific role is going to be networking with these groups. Starting with this group, we are going to have this thing. And Dr. Vishnupriya left. I think Vishnupriya left. Yes. Otherwise, yeah, I will ask her to prepare for this webinar next time. And Aparna, Aparna from Aparna, I'm not able to see her.
Dad, sorry, Dad.
Okay. She's my daughter only because she is in US. So I thought whether she is there because some other Aparna I have seen. Okay. Yeah.
So there's a comment by Dr. Lakshmi Chandravati. Thank you. It's been a very interesting session and it's a nice beginning. That's what she said.
Good.
Dr. Vani says, I joined late, but I shall, look forward to listening with you. And
Okay.
Dr. Rajalakshmi says she will coordinate with Dr. A.S. Kumar. Sharat Chandra would like contact information and facilities available and charges for the same. So could you please leave your phone number or contact Dr. Srilata? We have posted her number there.
Okay, good. I think good interaction and good beginning. And I must thank for the idea started by Professor Raghavendra Rao. For Genome Foundation, it is going to be a new dimension that you have given this idea, and we are going to continue this these webinars, monthly webinars. A decision is taken now, monthly webinars. We have to come up with the new ideas. And since we are forming a group here, we are also going to ask the group also to suggest different topics.
Thanks, sir. Thank you.
Topics so that we'll prepare. Genome Foundation will work hard to have these webinars arranged from our side. I think this group of about 100 people is going to be the ideal group. Anyway, we have thrown it open for on Facebook and YouTube also for those who want to listen, and also in future also that will be available. So I thank our own group from colleagues from Genome Foundation, starting from Dr. Raghavan. Dr. Rajalakshmi, are you able to?
Yes, sir. Yes, sir. Can you hear, sir?
Yes, now you are able to. Yes, sir.
Thank you very much, sir. I mean, all the presentations were very good, very interesting. Actually, and I had worked earlier for genodermatosis, sir, in SIG IADVL. And now, I mean, I'll be coordinating with Dr. A.S. Kumar, sir, in all the things.
Yes.
And one doubts what I had was, Bishwa's Amitabh has shown very well about the indications, clinical indications. Once last week, I had a patient showing that, I don't know why they have done just in an Imperial lab, I think. They have done just all the diseases, everything, all the mapping. They just go get the genetic disease, and they had come to me telling that whether there is a slight chance of getting vitiligo, I mean, whether I'm having vitiligo. But I don't know why, it has been done. So that was one question. And one more thing, sir, I just wanted, what are all the tests done and which labs it is done? If you make a list out, so it will be easier for all of us, like, to get all the tests whenever we suspect a genetic disease, like genodermatosis, we can go in for those tests where, so that it will be readily available, sir. So I would request, if we can get the list of investigations done, the genetic tests done, sir, in the labs. So these are the things which I really wanted to ask. So thank you, sir. Thank you for giving me an opportunity. Thank you.
Thank you, Rajalakshmi. I think your request is for specifically dermatology diseases.
Genodermatosis.
Yeah.
Yes.
Professor Raghavendra Rao will will take this because this is the purpose for which Dr. Raghavendra Rao wanted to have this webinar. He wanted to make himself known about his interest and his commitment and his contribution to this subject. And he is a avid researcher. And at this age also, he wants to always do something. I am sure that your suggestion will give lot of enthusiasm to him, and he'll be interested to do that. I request Raghavendra Rao to prepare such genetic tests related to dermatology available with Genome Foundation. Genome Foundation maintains the quality. Genome Foundation is not interested in doing tests for the sake of tests.
Yes, sir.
Which in this country, not in this country, which everywhere this is being done. We are not interested in that. That's why we do free counseling. We'll spend, where doctors cannot spend 5 minutes, we'll spend 5 hours with the patients. That's the advantage of Genome Foundation, because we have volunteers who are interested to help the patients, who are help the society, and help especially the people who are underprivileged. Most of the people are not able to even reach out to the people. So this is what the Genome Foundation's core objective is. I'm happy that you mentioned Dr. Kumar, whom I know for quite some time, and of course Dr. I.S. Reddy. I have visited him with my grandson, and likewise Diksha. I think she left just now. She was also instrumental in bringing a lot of our colleagues right from in Vijayawada and from other sites also. And I'm sure that the interest that has generated in this webinar will definitely take us further in making our objectives, all our objectives, combined objectives fulfilled as professionals. Thank you very much.
Thank you very much, sir. Actually, I mean, you mentioned about Dr. I.S. Reddy, sir. I learned a lot of dermatopathology from I.S. Reddy, sir.
I see, he's
we
there.
coordinate a lot.
He's still there.
Thank you very much.
At that time, for a young boy, when he was 4 to 5 months old, I brought him from USA, and almost for up to 4 years, I used to go regularly to him. So later on, of course, he left to USA, and then there was no need. Anyway, thank you for connecting again with him. Thank you, Dr. Kumar. And once again, I thank Professor Raghavendra Rao. Amitabh has given excellent presentation. Thank you, Amitabh Biswas. We would like to continue association with you in whatever manner that is possible. And we look forward to have contributions from Genome Foundation to every one of you. Thank you all.
Thank you, sir.
And bye-bye.
Sir, one comment, sir. Thank you, sir. Thank you for the nice presentations that we have received till now. We are interested to have this every month. You told us will definitely. I want to be a member of this, sir. Your permission, I want to join in this.
Absolutely. This is for the society.
Yes, sir.
The advantage of Genome Foundation is we are for people, by people, and of people. And for your information, all the funding for Genome Foundation or Truth Foundation is coming from my personal, not taken from anybody.
Oh, sir,
no loans.
Oh, sir. Nice.
No borrowings. No donations.
Sir.
Nothing out of societal interest.
Okay.
We are committed to the cause, and we would definitely be happy to be associated with all of you. And Bhanuja Rani, I think we heard, and Pariplavi has noted, and we'll definitely work together. Thank you.
Oh, thank you.
Thank you. Bye-bye. Namaste.
On the end.
Thank you, Dr. Gandhi.
Yeah, thank you. And Pariplavi, can we leave now?
Yes, thank you all very much. We thank all the Genome Foundation, thanks all the attendees who have taken time off on a precious Sunday evening to attend this session. It speaks much, it speaks volumes about your interest in not just the Genome Foundation or the subject, the fact that you've stayed back till your concern for the cause also. So we look forward to a very productive relationship where all of us join hands and are able to lend a helping hand to the common man out there on the street, in the village, wherever. So thank you all very much. And it's been a very productive day for all of us. We thank, special thanks to the speakers and to the team behind all the three speakers. Thank you all very much. Namaste. To all of
Thank you, Pariplavi. Thank you. Bye-bye.
Thank you.
Thank you, ma'am.
Thank you all very much. Thank you. Thank you.
Thank you, Pariplavi. Thank you.
Thank you.
Bye-bye. Bye.
Thank you.
Thank you.
Thank you, Pariplavi. Thank you. Bye-bye. Aparna, bye-bye.
Bye, Tata.
Aparna, bye-bye.
Bye, Tata. Bye, Daddy.
కనబడ్డానా నేను చూసావా
Marco, it's morning for us, right? So we were like unprepared to show our videos.
ఓకే నమస్తే
Namaste.
బాయ్ బాయ్ అమ్మా బాయ్ బాయ్ నాకు చాలా ఆకలి అవుతుంది కిందకి వెళ్ళిపోతాను ఇప్పుడు మరి
Okay.
Bye, Tata. I love you.
థ్యాంక్ యూ థ్యాంక్ యూ థ్యాంక్ యూ
Good, good.